just started recording the program so we can have it for posterity. I'm Steve Gilson with the Alliance for Southern California Innovation. Welcome to our Research Innovation Spotlight on Neuroscience. We're super excited for this particular program. This is our second installment of the healthcare or life science track we have here. We do this pretty much every month. We alternate life science and tech. And this month we have a great lineup of presenters. We're super excited. This is really at the Alliance. We are super connectors in the Southern California region. Our role really is to connect as many people as possible who can help support and nurture the innovation ecosystem. We are, we're a nonprofit formed about 4 years ago. A big part of our emphasis is connecting the great work being done at the research universities throughout the region. And by the region, we mean pretty much everything from Santa Barbara. Hi, Cheryl, that represents you, all the way down to San Diego. I got a bunch of folks there from San Diego down there, and as far east as Riverside, of course. And so we're just super excited to have a great representation today, actually, of different schools and universities that are represent kind of the wonderful intellectual property and amazing innovators and inventors throughout the region. So. This program here is to help connect you all together. It really works when we have research informing commerce and commerce informing research. So today, the presentations you will hear, we really encourage you all to ask questions, give feedback, talk about the things that your companies or your portfolio companies are working on that are of interest, maybe that can help inform some of the research going on here. So this should be a great open forum. The presentations will be hopefully about 5, 6, maybe 7 minutes. Then we'll have time for Q&A, and Leticia will lead all of that. Leticia is our moderator. I will introduce her in a moment. But for right now, I also want to say that we do this in collaboration with our fantastic partners in the area, Bioscience LA and Dave Whalen. And we're also, this event is sponsored generously by Alexandria Venture Investments. And I haven't seen Hallie on yet, but hopefully she will come on soon.
Hi,
oh, you are.
She's on.
Wait.
Good morning.
You're blocked. There you are. Sorry, I had my little participants window over you by accident. It's funny. So to let people in. So yeah. So anyway, Dave, you want to just describe a bit about what you guys do, and we're really super happy to have them. Obviously, the Alliance, we are a general technology and life science, connector, but Dave really brings this true life science expertise to there. So.
Perfect. No, thank you so much, Steve. Really excited to be here for our first official session that Bioscience LA is collaborating with the Alliance on. And great intro there. So I won't add much. But Bioscience LA is founded in 2018 as the independent nonprofit organization to really grow the innovation capacity for life sciences in the LA region, growing funding, growing space, growing talent, pulling it all together through messaging to grow awareness. And so we work on programs like this with the Alliance. We've launched everything over the past year, from a diversity-focused internship program called BioFutures to working on a venture fund with Wavemaker 360 Health. We are about to open up our collaboration space in Culver City, and all of that is about really amplifying and accelerating the work being done in the region to create more life sciences innovations. And so it's great to see many familiar faces on Zoom here, and some new faces as well. This should be a great program today. So thank you, Steve, and the Alliance, Andy, for bringing us in. Thank you, Hallie, as well, for your support, Alexandria's support. And I think, am I passing it over to Hallie to say hello as well, Steve?
Sure, yeah. I was going to say thanks again to Hallie. And Hallie, yeah, please tell us a little about what's going on at Alexandria.
Sure. Thanks, Steve. Thanks, Dave. Really great to see familiar faces here, and welcome everyone and presenters as well. Alexandria, for those who don't know, we're one of the largest providers of lab office real estate. Our headquarters is in Pasadena, and we have a very sizable presence in San Diego. And then on the investment side, we're one of the most active life science VCs. We have about $2 billion under management. And I think, what really drew us to supporting this initiative was, the kind of crosstalk amongst institutions, industry, and VCs to support some of this early translational research, to really create kind of an entrepreneurial support environment to help some of this amazing work get off the ground. So really thank you for everyone who's joining today, and really looking forward to hearing the talks.
Fantastic. Thanks, Ganali. And now it is my pleasure to introduce our either fearless or just amazing moderator, Leticia Toledo-Sherman. We just, she is an expert in this field and was really helpful and actually really critical in helping identify which of the projects that were submitted to us by the tech transfer offices for consideration, and getting the right mix of not just geographies, of course, but actually topics and amazing innovations going on. So Leticia Toledo-Sherman, Dr. Leticia Toledo-Sherman is the Senior Director of Drug Discovery for the Tau Consortium and Adjunct Assistant Professor of Neurology at UCLA. And I'll let her tell a little more about herself and kind of kick off this. She'll also lead the introductions of all of you, because we want to know who's in the room and have you all say a little bit about yourself. But first, I'll have Leticia say hi. Hello.
Hi, everybody. Thanks, Steve, Dave, Holly. This is a real pleasure to be here and introduce this topic today. It was really fun kind of going through the amazing, selection of programs. To be able to choose only one program, 5 programs was a little tough. And I hope I highlighted, a diverse set of ideas from Southern California in the agenda. I am Senior Director of Drug Discovery for the Tau Consortium. It is a family charitable foundation investing in really very much aligned with the goals of the Southern California Alliance, is to take programs from the academic setting and apply rigorous drug discovery enablement and move them to the point where they can actually be commercialized by, early investments, seed funding, Series A financing, but really create data packages that take those ideas into, a robust, assets that could be considered. I'm a medicinal chemist by training, so I have that kind of chemistry lens looking at this, but I have spent the last 15 years working in neurodegenerative diseases, Huntington's, Alzheimer's related dementias, and previous to that in oncology and antibacterial research. So again, it's a pleasure to be here. I thank you so much for giving me the opportunity. I'm going to pass it to the audience who's not presenting today, and the speakers will have a chance to present, to introduce themselves before the presentation. So I am going to start kind of in, I guess, in the third row with Clive. I'll just hopefully, yeah.
Hi. I don't know where I am in the third row. I'm in some sort of box here. I'm Clive Svensson from Cedars-Sinai Medical Center. I work on stem cells and neurodegeneration, the hospital here, and we have a regenerative medicine program that's focused on ALS and Parkinson's. Thank you.
And Martin Brenna.
Hi, good morning, everyone. I am Martin Brenna. I am from Kairos Ventures. I work in the scientific diligence team looking at life sciences opportunities for inbounding and potentially to go into diligence.
Thank you. Whit Ryan.
Hey, so I'm Ryan Witt. So I'm the Hub Director of UCLA's Biodesign Program, which is focused on new technology creation of UCLA Health, but I'm also a member of Techco Standards Los Angeles, periodic angel investor in Cross Healthcare, and previous was running a therapeutics company. So.
David Charles Pearson.
Hi, everybody. David Pearson from UC Riverside. I've been 35 years in the pharmaceutical industry, mainly at Novartis and then a row of startups. I'm responsible for the entrepreneurial programs and incubators at UC Riverside.
Great, thank you. Nick Edwards.
Hi, I'm Nick Edwards. I'm a venture partner at LA Therapeutics. We have a venture studio and we're focused on early stage investments and company creation. Internally, we have drug discovery platforms. I'm a neuroscientist by training.
Great. Jing-Chuan? Hello? Jing-Chuan, can you hear us? Oh,
You mean
thank
me?
Yes.
Okay.
I hope I
Ying
pronounced your
Hong.
name correctly.
I'm Ying Hong Bao, and I'm a venture partner of Viva Bio Innovator. We're part of Viva Biotech Holdings, and we invest in early stage therapeutic companies at seed to A round stage. And I'm representing the company on the West Coast. I'm involved in deal sourcing and post-investment management. Nice to meet everyone.
Great. I hope I'm pronouncing names correctly, so I apologize ahead. The next one it might be a bit of a challenge. Llewellyn?
Hi, good morning, everyone. I'm Llewellyn Cox. I'm a general partner at Mars Bio. We are the manager for the Los Angeles County Bioscience Investment Fund. We invest broadly across entry-level startup companies in the biosciences. I'm also CEO of Lab Launch. We are rapidly developing and operating quality, affordable lab spaces for startups in the Southern California area.
Great. And thank you. Matthew Buckard?
Thank you. So I'm currently the director at a facility called B Labs, which is actually based in Philadelphia, and do consulting work on the side for a company called R-Accelerate, which runs a lot of the development programs for Medici Ventures in the EU. Have a background in small and large molecule drug discovery and development, mostly on the CNS side, and then worked with iPSC platforms since 2009.
Thank you. Scott Livingston?
Hi, I'm Scott Livingston. I'm the Chief Executive Officer of Livingston Securities. We're a boutique investment bank and broker-dealer focused on three challenges: financing the nanotechnology revolution, changing the way that innovation is financed on Wall Street, and connecting people with their local innovation economy. And I'll be happy to connect with anybody who'd like to send me a LinkedIn. Thanks.
Great. Thank you. Pascal Crotet?
Hi, everybody. I'm Pascal Crotet, and I'm a senior associate at Rivervest Venture Partners. We're focused on both biopharma and med device investing. We've been around for about 20 years. Our primary office is in St. Louis, and we also have another one in San Diego. Nice to meet everyone.
Thank you. Andy Wilson?
Yeah. Hi, Leticia, and hi, everyone. It's Andy. I'm the Executive Director of the Alliance for Southern California Innovation. We're excited to have you all here today and appreciate all the work you do in our ecosystem, and looking forward to today's conversation. So once again, our warmest welcome.
And David Yang?
Hi, everyone. My name is David Yang. I'm an associate at APC. We're Silicon Valley-based generalist fund investing in tech and biotech companies with $5 billion under management. And my focus is on biotech therapeutic investments and also company creation.
Great, thank you. Aiden Aceves?
Hi, good morning, everyone. My name is Aiden Aceves. I'm an associate with Vita Ventures, a bicoastal investment fund managing $1.8 billion for investment in therapeutics. I have a background in machine learning and protein engineering, and happy to connect with any of you. Thanks for your time.
Thank you. Cheryl Eglander?
Hi, I'm Cheryl Milton-Linder. I'm the director of the Office of Technology and Industry Alliances at UC Santa Barbara. We have three primary functions. We manage the intellectual property developed at UCSB, including out licensing. We have a startup support program for our licensees and local tech companies. And if you fund research at UCSB, we have a team that will help get all the agreements in order so you can get started.
Great. I apologize. Something happened to my screen and you disappeared. So I hope I picked up the right place and don't skip anyone. Walter David?
Thank you. Walter David, I'm the Director of Research Business Development at City of Hope. And City of Hope is an academic research and Cancer Treatment Center. Basically, as my title says, I tried to take our research efforts and partner with an industry, also partnership with venture capitalists to form new company formations. Also, a new initiative that we have that I help manage is an internal fund to really kind of take our promising research efforts and move them down the value chain, making them more promising for would-be in-licensors and or new co-formation. It's a pleasure to be here. I'll put my contact information in the chat and feel free to reach out.
Great, thank you. Mark Springle?
Sorry, was on mute. Hi, everyone. Pleasure to meet you all. I'm Mark Springle, associate at Vita Ventures. My colleague Aiden introduced us earlier, but to echo, we're a Boston, LA-based venture capital fund focused on funding next generation therapeutics. I'm also a neuroscientist by training and looking forward to the discussion today.
Thank you. And Mary Raven?
Hello, my name's Mary Raven. I work as the Senior Licensing Officer in the Office of Industry Technology Alliances at UC Santa Barbara. So I work with Cheryl. I support Dr. Kosik's technologies and many other of the biotechnologies at UC Santa Barbara.
Great, thank you. Debbie Lynn?
Hi, everyone. Debbie Lynn, Executive Director at Boehringer Ingelheim Venture Fund. BI, it's a large top 20 pharma company. We're a corporate strategic fund. We invest in digital health, which I lead, and therapeutics. 500 million assets under management. We generally stay in the C to Series A stage. I have a background in bioinformatics and pharmacogenomics, and I've been with the company for about 15 years. Great to meet you.
Thank you. I'm probably not going to do a good job on this one, but Maria Kachuk? Hello, Maria. Can you hear us? Okay, maybe we can move on and give her an opportunity to introduce herself later. So Kishiro Yano. And then Shane Moise.
Good morning. My name is Shane. I'm with the Office of Innovation and Commercialization at UC San Diego. Great to meet everybody.
Okay. Maria or Kishiro, can you hear us? Okay. Well, maybe we can start to kind of get going with the program. Steve, is it the next? Basically, are we ready to go on?
Yeah, we're ready to go. And I can tell you that Maria is from the UC Irvine Tech Transfer. I know that, and Kishiro is an investment firm.
Great, thank you so much. I apologize. I hope I didn't leave anybody. If I left anybody off, please let us know and introduce yourself.
I'll say one thing, which is just a minute of housekeeping here, that if you want people to contact you, please do continue to put your information in the chat, and I will distribute that later. I just didn't want to just send everyone's contact information unsolicited, but obviously the real value here is to get everyone connected and make sure. Especially, please do reach out to the presenters afterwards if you specifically to get in touch with them. All that information will be made available to you. You can ask your questions, by the way, in the chat, but of course you will be able to also verbally ask your questions to the Prebys after the presentation. And from here, I'll just I'll sit back and be quiet and let you take over the show, and if anyone has any questions.
Great, thank you, Steve. So I thought I'll provide what might be obvious kind of introduction to the group gathered here today, but I think it's worth sort of touching on the fact that about 20 years ago, pharma exited neuroscience, just as the prevalence of neuroscience diseases started, and especially neurodegenerative diseases started increasing. And so there's been a void, and I think has become really clear with the approval of aducanumab from Biogen, how that has become a controversial drug, and how can we not do better than what that drug is doing. But it's really created an opportunity. And I think pharma is making a reentry. I think we see that on a daily basis now, small steps or large steps towards reentering into neuroscience or restructuring. Yeah, and just I think it's, again, common knowledge is about 6% success rate to develop a CNS therapeutic, and it's about $2.6 billion in research and development for each one of those CNS therapeutics that gets launched by pharma. So it's, you know, it's an expensive therapeutic area, and there are other ways that pharma makes a lot more money. You know, even after this 20 years, there's still I would say, dare to say, there's a fairly rudimentary understanding of these diseases. They're very complex. There's comorbidities. You know, the brain is a very complex, you know, organ, as you know, and that has really impaired development of drugs. And there are not that many, you know, drugs in pipelines and CNS pipelines, and therefore some pharmas have stayed strong and continue to make a headway in many areas of CNS, but for others that has been a void. So, you know, while all this is happening, I think there's been huge advances in technologies, right? Enabling technologies, CRISPR, you know, omics, imaging technologies that really have made this area a lot more tackable, more approachable. And I think that reflects why pharma is coming into. Even so, there really aren't that many targets. It's really surprising when I speak to people in pharma who are looking at, you know, acquiring programs or or investors, how the good targets are not that readily available. They look interesting, but, you know, the complete packages aren't there. So that's kind of where we are, I think. So it's a really good time to have this conversation and introduce you to these emerging programs. And I do encourage you, I don't know, Steve, if I think it was in the agenda, the rest of the programs that could not fit the agenda, but I encourage you all to look at them and see what fruitful conversations can be had around those. But today we'll start with introducing Dr. Ken Kosik. I have to say I have the pleasure of working with Ken as part of, he's a scientific advisory board member to the Tau Consortium, is an incredible resource. And I'm very fortunate to work with Ken and have his advice in neuroscience. And Ken was 2021 Potemkin Prize winner. It's a really amazing honor to have Ken here today, given his accomplishment. He's very important figure in the development of the understanding of the familiar Alzheimer's populations called the PISA mutations in Colombia. So I am going to pass. Ken is going to do, we're going to do a couple of tag teams because I think these programs have components of biology and components of chemistry. So Ken is going to introduce his work on Farnesyl transferase inhibitors for neurodegenerative diseases, Alzheimer's and Huntington's, potentially other diseases. And he's going to work with Steve Olson, who is his partner medicinal chemist out of the Burnham Institute in San Diego, and they will take on. I'm going to ask both of them, if I left anything out of the intro, to please highlight that in your introductions, and we'll start with the presentations. Ken, can you?
Yeah, so I'm going to share screen here. I hope I'm doing that. I'm pressing share. And
while you, I just wanted to make a housekeeping announcement. Please put your questions, if you have any questions, put them through the chat. I'll be monitoring the chat while Ken speaks, and I will be fielding questions at the end of his talk.
Okay, so if somebody can just give me a signal that you can hear me and see the slides. Is that the case?
Looks great, thank you.
Good. Okay, so thank you, Leticia. Thank you, Steve, for doing this. I'll just add to the introduction that I'm a neurologist and a neuroscientist. I co-direct the Neuroscience Research Institute at UCSB. worked on a lot of different things. Since we have an investor group here, some of you may be familiar with a little bit of work we've done on the Alzheimer value chain with Andrew Lowe at MIT. But really my work is, I'm much more of a neurocell biologist. And just as Leticia said, we've gone off after novel targets, and we've had some success in doing that. And therefore, about 2 or 3 slides in, I'm going to turn it over to Steve Olson, who has become an essential member of our team, he's a crack medicinal chemist. So let me just open with a quick background here. We're targeting these diseases. They're under the general category of tauopathies. They make up what are called primary tauopathies that you see listed there, in which the primary pathology is tau. And then there are the secondary tauopathies, in which the primary tau problem may be something else. In the case of Alzheimer's disease, amyloid, and in the case of CTE, head trauma, and that tau may be secondary. So we recently published a novel pathway that would lead, that's druggable and look very promising for clearing tau. It really targets an enzyme called Farnesyl transferase. And when that's inhibited, we get a very dramatic result here in both the behavior, in which we can see here that animals normally will chew up those little pads you see in panel B when you put it in there. But when the animal has a tau gene, when it's made to develop a tauopathy, they tend to ignore the pad, as you see in the panel B, compared to the control in panel A. But with the treatment of the drug, which is lenofernib, a well-known and FDA-approved inhibitor of farnesyl transferase, we can restore the behavior. Pathologically, you can also see down below in the images below that the image on the left shows that the animal has lots of green. Those are tau tangles, but the drug will clear it. And we have opened up a pathway by which we believe this occurs. That is, farnesylation will attach different proteins to the membrane. We believe that this protein called RES is very important. But the main point I want to make in this slide is that the mechanism operates by activating the lysosome. Ordinarily, this pathway will inhibit the lysosome, but by activating the lysosome, we believe that we can quite specifically clear mutant tau. So from there, I'm going to go over to Steve.
Thank you. So what we've engaged in is the discovery of second generation farnesyl transferase inhibitors, or FTIs. And the reason for that is that known FTIs, of which there are many, cannot reach high drug levels in the brain. And so when we measure the brain to plasma ratios, usually it's greater than 10. So our hypothesis is that all of the published FTIs, including ones that have gone into the clinic, are substrates for efflux transport proteins that exist on the blood-brain barrier. So ABC transporters such as P-glycoprotein or PGP. 2 of those examples are here. One is L778123 from Merck. The other is lenafernib, which was originally from Schering-Plough. It was recently approved by the FDA for progeria. But when we put these compounds into cells that overexpress PGP, we see very large efflux ratios. So we believe these compounds can penetrate the blood-brain barrier, but they're efflux right back out. And we also, in analyzing these molecules, we believe that each molecule has a functional group that is a recognition element for PGP. So our objective is to design novel FTIs that avoid this transporter-mediated efflux. So next slide. So we have found such compounds that, in fact, fairly rapidly. And so what we believe is this in vitro efflux assay can predict in vivo brain penetration for this particular series of compounds, and that we can achieve very high and consistent drug levels in the brain. So what I've shown here is a pharmacokinetics experiment where we measure plasma and brain concentrations over a 6-hour period, and you can see that the brain concentrations are extremely high and consistent with the plasma concentrations. So we believe for the first time we've identified farnesyl transferase inhibitors that can achieve high drug levels in the brain. We're continuing to work on this, and we've recently reduced or improved our potency to 18 nanomolar, and we think we're really on track to discover some novel therapeutics. Thank you.
Good. So, Leticia, if you'll allow me, just take one more minute to show you one other target that we've more recently identified. Do I have another minute or minute and a half?
Sure.
Okay, so we recently published a paper in Nature that found that, which we showed that the uptake of tau is mediated primarily by a receptor called LRP1. The spread of tau, just like tau behaves like a prion, in which it spreads through the brain according to this Brock staging. We developed a very simple CRISPR-I assay to look for candidates that will inhibit the uptake. And when LRP1 is suppressed, we can really completely inhibit the uptake of tau. You see LRP1 over on the right, and very closely related receptors do not do that. So we have been able to show that in animals, we can also inject tau and show that it inhibits the spread using a very nice construct in which in the animal, you separate the GFP, which stays local, from the tau, which spreads, as you see over on the right. But on the image on the left, tau is no longer spreading with the drug. And finally, my last slide is just to again talk about the mechanism here. We believe there's a very important nidus where a lot of the pathology occurs. That is, LRP1 is a receptor on cells that takes up tau. ApoE binds there too. You're familiar how important ApoE is in the process, as well as HSPGs. So we believe that tau as a transverse from cell to cell is somehow engaging in this complex. And when it engages in this complex, these various forms that you see in the lower right, these cryostructures begin to misfold and template misfolding tau in neighboring cells. So that's one of our future directions. A second really important target, which Steve is also helping us on now. We're in the very early days of developing tau-specific inhibitors of LRP1. I'll end right there.
That is excellent presentation. Any questions from the audience? Feel free to just sort of unmute yourselves and ask questions. I have a couple of questions, and I'll ask one while you guys are thinking of the next question. I think we have about 4 minutes, Steve. Is that okay? Okay. So, Ken, very curious about the Farnesyl transferase as a post-translational modification that is irreversible. And I'm wondering about what your thoughts about dosing, given that you might not need to have a sustained kind of target engagement, given that you have your farnesylation mechanism, and then the protein has moved on to another cellular location, subcellular location. So I'm just wondering if you've started, or Steve, to think about whether this is a pulsatile dosing regimen and how much of a compound you need to have on board to really engage that mechanism.
Steve, you want to take it?
I think that's mostly work in progress to know how much drug we need on board. I think in Ken's original study, the drug levels, because they did not get into the mouse brain very well, were relatively low. But we couldn't do a proper dose response because of the poor brain penetration. So I think that's still an outstanding issue that we need to understand. But I don't think we need to hit IC90 or anything like that, given the data that we have.
I had a quick follow-up question. This is Clive Svensson. Hi, Ken.
Hi, Clive.
Have you tried, because the transporters in the mouse, the BMECs are very different to the human transporters. I just wondered if you tried it in any human BMEC models and see if the drug gets across there the same way. Maybe some inserts or other technologies for that.
The only thing we've really done so far is some of the work that Steve has done on the MDCK cells. But nothing beyond that.
we should talk maybe because we've got some blood-brain barrier model on an organ-on-chip system running very nicely now, where we have human BMECs and neurons at the top. So it'd be fun just to put some through there and see if it got spat out in the same way from that transport.
It would be, yeah, Clive, I know you're in our neighborhood, so I'd always wanted to collaborate with you. So that'd be fun.
I'll be interested in learning more about that model as well for some of our programs. It would be fantastic.
I'll put a link in the chat box to the paper.
Great.
Non-scientific question, what's the cost to get this to the next milestone? And is that next milestone IND, or what's the next milestone, and what do you guys need for that?
Well, I think our next milestone is going to be to show that the compounds that Steve is coming up with will function well in animal models.
Yeah, I'll just add that I think that we want to establish a robust PD-PK relationship. And we do have a PD model that we're working on. And so I think that would be the next stepping stone. We are also applying for NIH grants to get undiluted funding to help move this program forward. So I'm not sure we can at this point give you a exact dollar figure.
So we have about 1 more minute. Any other questions? And I'll just make a comment that I know that Ken has been able to get a robust kind of IP package around the mechanism for Alzheimer's and related dementias, tauopathy. So I think this is that was a really good move to kind of identify the mechanism and then do a filing and really convince a patent examiner that is, even though the compound has been around, it's a novel indication. So I think that's a really, really good move on Ken's part that will enable the development of those compounds. Sorry. So I think any other questions, just please follow up on the chat message or by email as you're exchanging. So I think we will go on to move on to the next talk of today, if we're okay to move on, Steve. Good, okay. So the next talk is by Justin Ichida and by Sam Alworth from AcuraStem. So Justin Ichida is professor of stem cell biology and regenerative medicine at USC. His main interests are in stem cell biology and regenerative medicine, and he's founded AcuraStem. And today they're going to tell us, and again, please let us know more about your profile, Justin and Sam. He's going to tell us about PIK5 inhibitors. The title is a little bit different. Using human stem cell disease models and genetics to target convergent pathways and ALS. And I think he's going to tell us a little bit about the role of PIK5 inhibitors as potential therapeutics for ALS and other diseases. So please go ahead, Justin.
Great. Yeah, thanks, Leticia. Yeah, sorry to throw you the curveball. We are going to talk about PIK5 inhibition, Sam and I, and Sam's controlling. So if you want to go to the next slide, please. So in my lab and at AcuraStem, we use an all-human discovery platform to identify therapeutic targets for neurodegeneration. And one of our focuses is ALS, and it kind of makes the case for why this all-human discovery platform is important. ALS is representative of several, well-known neurodegenerative diseases in that its disease etiology is extremely diverse. So there are over 25 known genetic causes of ALS, and together they only explain about 10% of cases. So the other 90%, we don't yet know what genetic lesions they may have that are increasing risk for disease, but we do have evidence suggesting that a fair portion of those cases are actually heavily influenced by genetic mutations. And so if you really want to make a big impact on the disease, I think we need to find targets that can act broadly across these different disease forms without even knowing exactly what the genetic mutations are. And I think if we address that point, we will begin to overcome the problem that we've seen in the clinic, where we've usually been developing therapeutics for a very specific form of ALS, but then testing them in the clinic on patients representing many different forms of the disease. So if we go forward, what we try to do is integrate human patient-derived cell models, iPSC models, with human genetics to find targets that are both likely to be broadly efficacious amongst the different neurons that we see in different ALS patients, but also are likely to modify disease in humans based on the genetics. So if we go forward, to describe the iPSC platform a little bit more, what we've assembled is a large cohort of samples from different ALS patients. And this has now been expanded tremendously by Clive and his team for Answer ALS, in which they've got 1,000 different lines. But what we have are now, induced pluripotent stem cells from all of these individuals. We can make neurons, the neurons that are affected in ALS, as well as other glial cells that also have an impact on the disease. And what we're able to see is actually mimicking of many of the key disease hallmarks that we observe in patients. For example, faster neurodegeneration, which is shown here for patient lines versus controls, but also the protein pathology is similar, and a lot of the gene expression changes are conserved. So we're using the genomes of the patients in their blood cells, essentially transforming them into neurons and then using them to find therapeutics that rescue disease across many different types of ALS. So if we go forward, one of the most potent and broadly active targets that we found by screening across many different patient lines is inhibiting PIK5. So PIK5 is a kinase, and we decided to pursue it because it's genetically implicated in ALS, because the phosphatase that acts opposite of PIK5 can cause ALS when it carries loss of function mutations. So essentially, these mutations that do the opposite of what we're doing can cause ALS. And so what was interesting about PIK5 also is the mechanism is quite intriguing. So inhibiting PIK5, we found actually slows the progression of normal autophagy, which is the fusion of autophagosomes and lysosomes to get rid of misfolded proteins from neurons. And by doing so, it activates an unconventional method of getting rid of these proteins called secretory autophagy, where these misfolded proteins are secreted from the neurons and they're scavenged by surrounding microglial cells. And this is incredibly efficient at getting rid of these misfolded proteins. Importantly, because that is a common nexus of neurodegeneration, these misfolded proteins. This works very broadly across the types of ALS patient neurons that we've tested. So on the right, you're looking at, we can see that inhibiting PIK5 can lower the likelihood of neuron death in all of these different ALS patient lines, from C9 to sporadic ALS, as well as ALS caused by FUS and TDP43 mutations. And so what we've done is we've actually checked, does this secretory autophagy mechanism, is it applicable to other types of misfolded proteins like tau, for tauopathies? So in collaboration with Sally Temple and Alison Goat, we've generated organoids, brain organoids from people with FTD that have tau mutations. And you can see that on the left side, that suppressing PIK5 can actually rescue the survival of these patient neurons in these organoids by getting rid of misfolded tau. And so although we use humans for discovery, when there is a mouse model that is applicable here, we do use it to finally test our approaches. And here we have a TDP43 ALS FTD mouse model, and the mouse on the left was injected with an antisense oligo suppressing PIK5, and the one on the right was a negative control. You can see the mobility is quite different, and PIK5 suppression extends the lifespan by about 25 to 30%. So I'll hand it over to my colleague Sam, the CEO at AcuraStem, who's developing therapeutics for PIK5.
Thank you, Justin. Yeah, it's a pleasure to be here today and tell you about how we are developing antisense treatments for PIK5 and other targets. So with my background's in bioinformatics, machine learning, so at AcuraStem we do have a good infrastructure for bioinformatics. And so we've developed our own software, proprietary software for designing ASOs. So it enables us to create every possible sequence for the gene. And based on certain criteria for quality that we have, we can prioritize certain sequences. We can also look at where they might suppress elsewhere in the human genome, and we can rank them based on their off-target prediction. And so then we synthesize the most promising candidates. And then, as Justin mentioned, at AcuraStem, we also have a large panel of ALS patient lines, and we can do the in vitro efficacy evaluation of different antisense candidates in terms of survival and other phenotypes. And that's also enabled by proprietary algorithms and machine learning models. We also look for potent suppression in the human neurons, and then we move into a humanized PIK5 mouse model that we've established. So we prioritize leads that have efficacy and suppression in human neurons, potent suppression in vivo without glial activation in the mouse model, and no off-target safety concerns. And we've assembled a great team at AcuraStem for this development of the PIK5 and also our second program, which is just 6 months behind. As I mentioned, my background's in bioinformatics, machine learning. Justin is a leader in disease modeling. Dr. Chang was a postdoc fellow in Justin's lab, and she's our head of research, and she runs the neuron platform and histopathology and biomarkers. Peter Cesani is a consultant who brings immense ASO development expertise. He was at Sarepta, where he developed Tleperson from preclinical all the way through to new drug approval. Jim McCardle is an expert in chemistry, formulation, quality control, manufacture, formerly a VP at Ionis. So we have a development candidate, AS202, which we selected from hundreds of sequences. If you look on the left, it's showing the efficacy in human neurons. If you focus on these first 3 bars, this shows the hazard ratio compared to the healthy control. So without treatment, the patient line shows a high hazard, and then with treatment, it shows less hazard or increased survival. And this goes down in a dose-dependent fashion with treatment. A high dose is not toxic in control neurons. Moving into the in vivo human model, we look to see potent suppression of PIK5 without peripheral knockdown in the heart and other organs, and then without glial activation of these glial activation genes. So I think we might be over time, but we definitely want to have time for Q&A. I want to thank our funders, especially Viva Biotech and Ying Hong, who's in the audience. And we've also had tremendous support from NIH and Department of Defense and patient foundations like the Alzheimer's Drug Discovery Foundation. Thank you.
That's great. And we can move, we have about a minute of questions, and of course, continue the Q&A on the chat and by email. Any questions? I'm curious if you could just touch about potential like safety issues with PIK5 inhibition, if that's something that you've looked at, mechanism-based talks.
Yeah, so it turns out that there are people, mice and humans that have a loss of function copy of PIK5. So and they're healthy. So we know that knocking out 50% of PIK5 activity is completely tolerated, and which is also sufficient for the therapeutic benefit. And there are some animal studies showing that they don't see toxicity until about 90% inhibition of PIK5. So I think we have a window there. that we feel is suitable for targeting PIK5. Yeah.
Right. Well, thank you so much. This is really excellent. Again, I encourage the discussions to continue in the chat or directly. We're going to move on with the program, and we're going to have Dr. Seema Tiwari-Woodruff from UC Riverside. She's professor of biosciences. She has an interest in MS and understanding myelation, remyelation, and especially looking for drugs that can delay the advance of MS by remyelation processes. And she's going to tell us about alleviation of extensive visual pathway dysfunction and remyelation with a remyelation drug. And please go ahead, Seema.
Hello, everyone. As I was introduced recently, I'm Seema Tiwari-Woodruff. I'm professor at the University of California, Riverside. I also wear a couple other hats, including Director of Biomed Sciences, and I am the President of American Society for Neurochemistry. You all are welcome to look up the society and be part of it. All right, so digressing from the title, I'm going to actually give you an overview of why we think that ER beta ligand is actually a good drug. So when you look at multiple sclerosis, it's basically got over 1 million people in the United States who have MS at this particular point. It's a neuro degenerative and inflammatory demyelinating disease. So it has a lot of symptoms, and the disease is not focused on one thing. And as you can see, the number of problems which people have, so you actually have to look at decreasing disease symptoms holistically, not just one way. So one of the things I want you to focus on is the vision problem. More than 40% of the patients have vision problems, and you can actually use this because you can test this in animal models, as well as humans, as a therapeutic biomarker. So you can look at the disease progression, and if you have a therapeutic drug, you can actually see alleviation or changes. So when a patient walks into a doctor's office during MS, there are quite a few things which are happening. One of them is inflammation and regeneration has been ongoing, but the patient really doesn't know this. It's only when they actually have paralysis or eye problems or urinary problems, that's when they go to the doctor. They go, okay, something's really wrong with me. By that time, the red circle shown here, this is the time when they see the doctor. It's the degeneration process has started. So demyelination and axon degeneration is ongoing. And this is a big problem with most of the neurodegenerative diseases. When the patient walks in, we can't do a preventative mechanism. It's always trying to fix things which are going wrong. So in the case of MS, most of the drugs which are present, which is shown on the treatment side on the right, are immunomodulatory. And there are no drugs which actually directly affect the CNS and fix the problem which is ongoing. So everything which is happening in the CNS is indirectly happening because you are suppressing or you're modulating the immune system. So there is an unmet need for finding remyelinating drugs. And as shown in the bottom half of this slide, there are quite a few clinical trials which have been processed, and some of them have failed and others have sort of like digressed, and some are still ongoing. And the results have always been not conclusive. And there are many reasons for that. But when you look at a patient's life, the MS disease is mostly women, which are very young. If they are pregnant, what you find is during pregnancy, they actually see a decrease in relapses. And the question becomes is why? So it turns out that when a woman is pregnant, there are quite a few things which are changing. One of them is, of course, an increase in progesterone and estrogen. So the particular estrogen which goes up here is the estriol, is the pregnancy hormone, and it affects both ER alpha and ER beta receptors. So the receptors which are affected by estrogens are many, have many functions. So estrogens are important for reproduction, fluid intake, learning, stress, eating, and maintaining a CNS milieu as well. So if you look at ER alpha, it's actually a breast and uterine cancer causing receptor, and also it causes feminization. So when you think of women who have a preponderance of having any kind of cancer, they cannot take this drug. And of course, males can't take this drug. So long time ago, we discovered that ER beta actually, ligands actually managed to treat the mouse model of MS and produce remyelination. And I'm just going to give you an example here. This is showing an electron microscope image. The top row is normal. The second row is when you have disease. So you start seeing demyelination. The black circles around are myelin, and you can see that there's a lot less in the second row. The third row is pre-treatment with the ligand, and you can actually see that there is more myelin in these axons. So what we did was we just did not want to look at myelin in the concept of how beautiful it looks. And this is one of the biggest problems we've had in this field is people don't do functional analysis. Myelin's job is to increase conduction. So we do a lot of I have an electrophysiology background. My lab does a lot of electrophysiology to show that when you have remyelination, conduction changes. In the meantime, I met Dr. Castleman-Bogan, who actually is an estrogen specialist. He said, we need to collaborate. I have ligands which are more superior than the one you were using, and let's try them out. And that's how my collaboration with Dr. Castleman-Bogan started. I'm not a chemist. He's amazing. His lab changes side chains, side molecules, and makes these efficient ER beta ligands. And we have published really well. And recently we got an NMSS Fast Forward grant to do one step forward to do the PK studies and toxicity studies, which we are doing on various different ligands. One of the things which is really hard to explain is people don't do the right studies. That's why a lot of clinical trials fail. Because when they go from mouse to man, they don't really look at what's really happening with these molecules. So you really need to understand how they work, what the mechanism of action is, and are they influential in an environment which is in vivo. So you can do all the studies you want in cell culture and look really good. But when you go in vivo, you have different cell types, you have the immune system, you have damage. And how do these molecules behave? Do you still see that effect? And once you start doing that, then you'll realize that some of your compounds work really well in the cell culture, but they completely don't work when you go in vivo. So anyhow, we do many assays. And what we have seen is that if you do many assays, you can actually de-risk drug development. And I know I'm going really fast, but what I wanted to show you was that the tool compound which we have used in the sense indazole chloride, we have analogs of these, and they show very similar results when we look at disease, where we see a decrease in EA disease. We see conduction activity, so we see latency decreases, meaning that more myelin has been formed, and therefore conduction is better. We also do rotarod activities. What we've shown in the past is that these drugs actually stimulate endogenous oligodendrocyte pool. We also know that it works through the second messenger signaling, which is really conducive for keeping the cell survival and decreasing cell death. So one of the key things about MS is the environment is very deleterious. You have ongoing inflammation, and if you don't touch that, bad things can happen. You can't suppress that either, because inflammation has a good role as well. So what we noticed with our compounds is that we see a modulation of the immune system. The pro-inflammatory cytokines hang on, except for IFN gamma, which goes down, but we see two key chemokines which change. CXCL10 is a deleterious chemokine which actually kills oligodendrocytes. It's in part of the apoptosis cycle. CXCL1 is the opposite. So with these drugs, CXCL10 is suppressed and CXCL1 is activated. And that is a key thing. So you modulate the immune system and you initiate remyelination, which lingers and stays. And that's how it gives you those beneficial effects. So while we were doing these studies, and like 3 years ago, we started testing novel indazole analogs. So John's lab makes these compounds and hands it to us. We run them through primary cell cultures of oligodendrocytes, where we see if these analogs kill the cells or if they produce more myelin for these differentiating oligodendrocytes. And then we picked the best ones, and we did that for 2 compounds and took them into in vivo. And what we see is they were actually a little more superior than the tool indazole chloride compound. And what you see is this is the O-methylene O-chloro series. And pictures over here showing you in red is myelin and green is neurofilament. This is in spinal cord sections in the dorsal column. And what you see is there's more myelin being laid out when these compounds are present. And we published this in 2019. We again showed that the conduction is better. So, and they stay on the rotorod better. So this compounds actually affect function as well as overall health of the mouse, and showing you myelin over here. And again, we went through a series of chemokine cytokine analysis to see, we don't want the immune system to be suppressed, but we do want it to be modulated. We again see that IFN gamma goes down. We see CXCL10 really gets down from vehicle, this is vehicle, and then CXCL1 is upregulated. And as a control, we do pre-treatment and post-treatment with estradiol, which affects both ER alpha and ER beta. But these compounds, which are very specific to ER beta ligand, ER betas, sorry. So anyhow, bottom line, we have a potential to provide unique therapeutic benefit to patients with these compounds. They have very low toxicity. I don't even know of any right now. And because MS is a multifocal disorder, we need to have a compound which is going to affect different aspects of the disease. And in my lab, we have done studies on optic neuritis, EAE-related or MS-related optic neuritis. We've looked at cognitive dysfunction. We've also looked at the cerebellum and, basically motor cortex function as well. So yeah.
Seema, I'm so sorry, but we need to wrap up in about a minute.
Yeah, this is my last slide. This is my last slide. So what I wanted to end with was we have over 100 indazole chloride analogs, and we have the grant to study, conduct ADME PK studies on indazole. And we have inventors are available as consultant advisors to companies interested in developing this project. And that's it. Thank you.
Thank you so much, Seema. Unfortunately, we don't have any time for discussions, but because we have 2 more talks, but I do encourage you to please, use the chat button and communicate through that, or just, direct emails. Thank you. And we're going to move on to a little bit of shift from a therapeutic modality to technology, which is, I think, we're going to be, learning from Sunil Gandhi, who's developed this interesting imaging technology that I think is going to be really fruitful in understanding disease and potential target engagement and biomarkers. So Sunil Gandhi is Associate Professor of Neurology and Behavior at UC Irvine. And I'm going to pass the presentation to him.
Okay, thank you so much for the invitation to speak here. Let me just make sure you all can see this and in the right mode. Okay, I hope you all can see it. I'm having some computer problems. Just shout out if there's a problem. Okay, so again, thanks for the invite. Today, I'm going to tell you about some new imaging technology we've developed in the lab that we believe can dramatically enhance how we can locate drug effects in the CNS for preclinical studies. So today we've heard about a number of novel brain therapeutic possibilities. At some stage, almost all of these programs have to contend with the challenge of finding drug effects or target engagement at cellular resolution deep in this tissue, the rodent brain. Okay, and classically to do so, we've cut the brain into thin sections and placed those sections under a microscope. But what we lose is, of course, the three-dimensional structure. So new technology has emerged to preserve the 3D organization of brains, and that is by making the tissue optically transparent, like you can see here. And then what we can do is use a sheet of laser light to image the intact brain at very rapid speed. And my team has partnered with Zeiss microscopy to do just that. So what does it look like when you can peer into an intact brain? Here, I hope you can see a subsample of circuits revealed by our technology. The balls you see are individual cells. The brushes are the dendrites, and those cables are the axons. Here you'll see the individual descending axons of the corticospinal tract. This technology is, of course, makes for pretty movies, but it can do much more. And really what I want to leave you with is this idea that it can detect patterns in the brain that really no other tool can. Okay, so we have been building a suite of tools and technologies that can take you from whole brains to actionable data. And so we've developed tools that let you take a brain that has experienced some treatment regimen, make that brain optically transparent, label it with probes, and then image it on the microscopy system that we've developed with Zeiss. But then perhaps most importantly, we have built machine learning tools to detect features across the entire brain completely and automatically, and adapt. We've also adapted powerful statistical frameworks from RNA sequencing that can quantify where things happen on the brain with just a completeness and statistical rigor that is unmatched. So we've been talking about brain pathologies. Here is amyloid deposits from the 5X FAD mouse model for Alzheimer's disease. In green, you can see the plaques. In red, you see the closely associated microglia. So if you look on the left panel, I think what you'll notice is that in this cleared tissue, the pathology is pretty heterogeneous. It's broadly distributed in all kinds of patterns. How can we precisely quantify this pathology in 3D? What we have done is develop the tools to segment out each and every plaque in 3D, and then use the Allen Reference Atlas to automatically count the location of all of those plaques. And what we end up with is a complete summary of the pathology across individual mouse brains, which I would argue can detect patterns of drug effects that thin sectioning can't. Okay, we know we'd like to be able to monitor the activity of the whole brain at cellular resolution. Right now, the only technology that lets us do that is shown here. That is to visualize the expression of gene products that are intimately tied up with neuronal activity. And what I'm showing you here is a panel of mice and their brains after the mice moved from the dark and into the light. And it's not surprising, perhaps, that most of the areas that we detect responding are visual areas. But what's really cool is we've begun to find areas that we never would have expected. And so you can see the kind of sort of shape of the analysis tools we're using are reminiscent of sequencing tools. And what's really interesting are the new hypotheses that get generated by some of this technology. Okay, we've been talking about cell therapies in the brain. I just wanted to show you this movie of a mouse in which all of the endogenous microglia have been cleared away and replaced with human iPS-derived microglia. And I guess to deeply bury a lead, we have a NewCo that's developing this technology as a cell therapeutic platform. Okay, so to push on this idea, that I've been sort of hinting at, that we can quantify things that thin section histology can't, that we can detect patterns that might otherwise be missed. I wanted to show you this published example in which we looked at mice that had been infected with a parasite toxoplasma. And like many other neuro-focused parasites, the infection results in an infiltration of monocytes from the blood into the brain. Which has pathological effects. In previous literature, no one had really found detected a consistent anatomical pattern to this infiltration on a regional level, but we were able to discover, using these tools, one pattern of infiltration specifically into the olfactory tubercle that actually gave us really good new hypothesis for explaining the neural mechanisms by which toxoplasma disrupts the behavior of infected organisms. Okay, so I'm going to, I know we wanted to reserve time for questions, so I just wanted to acknowledge the people who have been involved in this work and our funding sources, and stop there and see what questions you may have.
Thank you, Sunil. That was beautiful.
I'm glad you were able to see anything because my computer's acting up. So good.
No, just beautiful, beautiful, clear images. Thank you. Are there any questions from the audience?
Oh, sorry. Go ahead. I see a hand raised.
Hi, really wonderful images. I had a question for Sunil. So when you are actually defatting the brain, right? You're actually to see the images. So what happens to the myelin? You actually was thinking about how wonderful it would be to do something like an MS model, because you will be able to see lesions and immune infiltration, which would be really beneficial when you're doing therapeutics.
Great question. Yeah, we have done a little bit of work looking at myelination. The procedure that we're using that I showed you for clearing the tissue is a multi-stage procedure which allows for us to bind proteins generally as our probe, and so we've used myelin basic protein as a target. So even though the delipidation, clears away much of the lipid structure, we nonetheless can have a signal to detect. So I see no obstacle to being able to use this to look at myelination and detect demyelination.
Thank you.
Is this commercially available yet?
Yeah, so the company, so we are funded by the Brain Initiative to disseminate this technology to the research community. And so we have some products that are already in the market. For example, the imaging platform that we've co-developed and co-marketed with Zeiss. We also have some other products that are just in the stage of rollout and some in product development. So that's this spin-out company, Translucence. But the overall goal of the company is definitely to enable the research community to do these types of imaging experiments.
Thank you.
Any other questions? Sunil, I'm curious if you've looked at any of the tau mutation models and to see if you actually can see the fibril structures, the neurofilament structures.
Great question. That's near the top of my list on the to-do list. We have not looked at tau specifically, but I don't see an obstacle to it. I think that just with some little bit of work, I expect that we should be able to detect it, just as we have looked at, obviously, amyloid, as I showed you earlier, and alpha-synuclein, which we've done quite a bit of work on. What's really nice about the alpha-synuclein work that might be relevant to tau is that we've been able to see and use the machine learning tools to dissect out different aspects of the alpha-synuclein pathology, where we can separate fibrils from some of the cell body expression. And so the machine learning component to this is really sort of enhances the power of this whole platform.
Great, that's excellent. Any other questions? That's just really fascinating. Thank you.
Maybe just a quick one. What's the throughput, especially as you're thinking about commercializing this, to image, one brain, like what does that process look like in terms of time and FTE intensivity?
Yeah, great question. So in terms of individual, so the whole process for a whole brain, rodent brain, soup to nuts takes between 10 days to 2 weeks. It's much of that is sort of waiting for incubation with probes. But the actual imaging time can be quite fast. And that's sort of one of the important limitations on our throughput. We can image a whole mouse brain with the kind of resolution that you saw in that image, the movie of neural circuits, in about 40 minutes. So we have, we regularly work in batches of 10 or 20 animals, 10 or 20 brains at a time, but definitely we're thinking about how to scale that up for internal production. But the other way to scale it up is to disseminate and democratize and get this in a lot of people's hands.
Great. Thank you very much.
Sure, of course.
Just fantastic. Thank you so much, Sunil. So our next talk is sorry, one second. I am having a problem with can you hear me well?
I can hear you well. This is Chad.
I'm getting a stability signal across my screen, so I apologize if my Wi-Fi seems okay. So sorry. So Chad Bayer from Versapeutics is going to present on a disease-modifying approach to treat Alzheimer's disease. This is a technology that's based out of Professor Yimin Zhu at UC San Diego. I'm going to let Chad has a stellar career in neuroscience and has quite a few of the drugs that you see on commercials probably to his name. So I'm going to let him continue with the intro and go on to give his talk. Thank you so much.
Wow, that was quite an intro. Thank you. Now I feel like I have a lot to live up to.
You told me this, so it's
it's there. Yeah, I know. So as Leticia said, my background is in neuroscience. I've spent the last 25 years in drug development. I like to say I've worked with 70,000 people at Big Pharma, and I've worked with 7 people in the last decade of a couple of smaller companies focused on CNS products. So I've met Professor Yimin Zhao over the summer, so I'm going to have a disclaimer that I'm very new to Versapeutics, joining as a consultant as we're bringing together an A round of investment and looking to explore wider opportunities within the company. I mention that because if you have really difficult questions, I can just sort of sidestep them and not answer them and say I've not been with the company long enough to answer. But I'm half kidding there. But it's a pleasure to be here, and thank you for the invitation. And you guys can see my screen, the slides just fine.
Okay.
Yeah.
Perfect. I'm not going to waste much time talking about Alzheimer's disease to this group. And Leticia mentioned it at the beginning, at the outset. The Biogen approval has made this area white hot again, and certainly maybe people are pivoting back to neuroscience. But what I also think the Biogen drug has done, it's reminded people of A-beta, and the fact that some people are just fine with some A-beta in the brain in terms of some of these plaques, and others are not. And so it's difficult to decide which camp you're in, that you've got to get rid of all these plaques or that you can live with some of these plaques. Some people argue that they're helpful in some normal brain function. And Yimin's lab at UCSD is really focused a little bit on a different approach and looking specifically at A-beta toxicity, how amyloid beta, how the oligomers destroy synapses, and particularly well-formed, well-functioning synapses in the hippocampus, how that synaptic loss occurs. That's really the foundation of what I want to talk to you about and the therapeutic that we're starting to look at. And so if I can maneuver, that worked, right? So the pathway that we're focused on is not a new pathway. It's a very, very important pathway. In fact, it comes in two different flavors. The pathway on the left, which I've grayed out on purpose, is a very well studied, been around for a long time, Wnt signaling pathway that, in fact, I don't know, 15 years ago at Wyeth, we had a Wnt signaling program as well. So this has been around for a long time. I think what we know is that in either case, whether it's the older, well-studied canonical Wnt pathway or this more, this different non-canonical Wnt pathway that we're focused on, they're very important for cell functions, both of them. So you don't really want to monkey around or tinker around in some of these components of this pathway. Instead, what Yimen's research is really focused on is regulating these pathways, and in particular in disease states. And so there's a lot of work on this particular slide that is probably close to 2 decades of his research, really discovering how these pathways assemble and how important this particular pathway, or rather target RIC, regulates the balance of synapses. And so RIC is very important for allowing synapses to form, very important for these pathways to maintain communication at these very important junctions in our cells. And this balance becomes turbulent. It becomes dysregulated in Alzheimer's disease. This has been shown in preclinical settings. What happens is amyloid beta through RIC causes an imbalance in this particular pathway. I don't know if you can see my cursor or not, but I'm pointing to the synapse disassembly and the out of balance and impaired balance that occurs. And what the team has really focused on is looking at RIC as a target. And as I like to tell Yimen, that this is great when you have a cartoon or you have a knockout, a conditional knockout animal. But what really is interesting, I think, is when it happens in humans. And this is exactly what does occur in the human pathology. One of the maybe strongest genetic pieces of information that occurs in Alzheimer's disease patients is a reduction in an endogenous RIC inhibitor known as WIF1. And so the idea being that even in patients, you see a reduction in this balance. The pathology removes this important regulator of this non-canonical pathway. And it really kind of confirms the idea that if you can restore the balance, particularly in the hippocampus where RIC is expressed, it's expressed at very low levels outside of the brain, expressed a little bit in other areas of the brain, but the densest expression of RIC is in the hippocampus. And that's what we're focusing on. And this is really the foundation for the program. Again, it's 15 years of research out of UCSD and Professor Yimen Zhao's lab that we just jumped over in about 4 minutes. So I'm sure I left out a detail or two, but it is the rationale for why we want to develop a RIC inhibitor. We have a multi-pronged approach, a couple of different antibody approaches. The most advanced and lead antibody is our Versamab 101 antibody that's been developed, that's proprietary, that has been shown to do exactly what you'd expect it to do. And I'll show you some data in just a second. This is a high affinity, so it's in the picomolar range. It comes from a suite of other antibodies. This is the most potent, the most active antibody. I think we tested up to 15 of this particular, of the 101 series. And I guess I would say that we are looking also at some small molecule approaches to this pathway. The most advanced is this particular monoclonal antibody. To date, what's been shown, I think, is very interesting. So the team has been able to look at these 5X FAD, this familial Alzheimer's disease mouse. It's a very aggressive, very severe form of the disease in a preclinical model. And much like you'd expect, gosh, I hope you can see my mouse cursor because it does get a little confusing. I'm looking at the left side, looking at total synapses, the number of synapses. And what you see compared to controls, just looking at the blue bar, whether it's total synapses on the far right of this particular graph on the left, or it's the pre or post puncti, you see a significant 40 or 50% loss of synapses in these Alzheimer's disease animals. So that's consistent with what you'd expect. And what you see is that when you treat the animals for 2 weeks, once a day with the monoclonal antibody, the VMAB 101, you see a complete restoration, a protection of these synapses. That's on the left side, which is great that the science that we started to talk about kind of pans out. You've preserved and protected some of the synapses. He's gone on to further look at LTP. These are functioning synapses that's data that's not shown here. So you're not just protecting synapses, you're actually protecting and they're functional, they're working. And I think a good example of that is shown here on the right side. This is a novel object recognition. This is looking at recognition memory. It's a very well-studied, well-used in vivo model of Alzheimer's disease memory. And what you see is not a surprise. You see that these 5X animal of a model of Alzheimer's disease absolutely lose their recognition memory. That's this third panel right here, where they do not explore this novel object like they did in the controls. So maybe I should work left to right. This is the control animals can remember the old object, spend more time sporadically exploring the new object. Animals that have the Alzheimer's disease do not. And the monoclonal antibody, again, given for 2 weeks, restores the ability of these animals to have this recognition memory. We've gotten a little bit further in the development of this antibody. We've had past funding. I should at least stop and acknowledge the support of NIH as well as Viva in bringing and progressing some of this research. We've generated master cell banks. We've started some of the IND enabling talks and GMP work. We can produce this with a CRO. We are, like a lot of the companies here, I think, really semi-virtual. So we rely extensively on CROs and labs outside to do the development work. So we've partnered with some world-leading CROs to make the antibody, but also test it. So we've completed a non-GLP tox study, both in rats as well as in non-human primates. My biggest concern, I'm sure it's already been asked as a question today, is toxicity. Are you concerned about toxicity, mechanism-based toxicity? And the short answer to that is yes. We are always concerned about that. But at least initially in our dose range finding studies in 2 species, we've not seen any evidence of toxicity. This is very well tolerated. Knock on wood is a good sign, but we are gearing up to raise money and continue toward IND. And that's kind of where we are now. We're in the process of constructing our Series A financing. We are, and the goal of that will be to advance the 101 antibody to IND. About half of that money is to bring it to IND. The other half of the money is to do phase 1 safety and tolerability in humans, as well as establish this important PK/PD relationship. So things like EEG and other biomarkers that we'll build into the phase 1 program, all with an eye to targeting demonstrating that what we've seen so far in animals actually recapitulates in what we see in humans. So I'm going to stop there in the interest of time and answer any questions if we have time for it.
We have about 1 minute, or Steve can tell us how we're doing in overall time.
Yeah, I mean, we're a little over time, but of course, if anyone has a question, if you can all hang on. And sorry, we're running a little late on this one, but there's so much good presentation, and so please go ahead if there's a question, please.
Wow, no questions. Perfect.
Do we have a question? I have a question, really quick one. So I was trying to figure out what kind of synapses are being improved. Is it excitatory, inhibitory, post, pre?
Yeah, it's a little bit of both pre and post, but largely what's been looked at so far is glutamatergic synapses. So the excitatory variety, that's been the bulk of the work is focused on glutamate.
Okay, and you guys did the LTP studies to show that those are enhanced.
Yes, yeah, and preserved, and there's also a conditional knockout of this pathway, because if you knock it out from birth, the animals don't do so well, not surprisingly. But a conditional knockout also confirms the LTP work as well as some of the work that I just described with the monoclonal antibody.
Thank you.
Yeah.
Well, that was amazing. Thank you so much for all the speakers for your really inspiring and you know, exciting talks with a lot of prospects for advancement into commercial, you know, entities. So I thank everyone. I'm gonna pass it back to Steve. This has been a pleasure for me to participate in this, you know, fun venue today. So thank you so much.
Yeah. Hey, everyone. Thank you so much for attending. Leticia, great job. And there's you know, so much content to go through, and you've done a great job managing us. We're just a few minutes over, which is, you know, expected sometimes when these things happen, these live events. So it's great. Our next spotlight will be on October. Sorry, I just had it in my head, but it's on immunology. I'll send out an email. It's like the second Tuesday. I think it's the 15th, and as well, we are recording this. I will post this video after in case you miss anything. Chad, I don't know if you have. It's okay to send around your deck. I was asking if you want to share if you have
Yeah, I could send it to you. Yeah, there may have been an update or two.
Okay, great, fantastic. Yeah, and we'll make sure there's information for everybody, all the different presenters. Make sure that's all shared, and of course, anyone who shared their information here will be in the follow-up email. So thanks to everyone. We look forward to seeing you again in October if we're gonna be around.
Thanks.
Great, thank you.
Thank you.
Bye, everyone.
Bye, everybody. Nice to meet you all.
Thank you all.